After studying this module, you shall be able to know about the types of sweat glands and the composition of sweat according to the glands, about the biological characteristics of fecal matter. Sweating is a process in which fluids are secreted by the sweat glands in the skin of mammals. This process is also known as perspiration or diaphoresis. Sweat is produced by the sweat glands. These glands are also known as sudoriferous or the sudoriparous glands. These words are derived from the Latin word sudor, which means sweat. These glands are small tubular structures present on the skin. These glands are a type of the exocrine glands. Exocrine glands are those glands which produce excretion, secretory products, anatomic distribution and distribution across species. Primates and horses have armpits that sweat like those that of humans. Although sweating is found in a wide variety of mammals, relatively few, with exceptions including humans and horses, produce large amount of sweat in order to cool down. Next is fecal matter. Excretion is a process in which the human waste is thrown out of the body or excreted out of the body. The fecal matter or human feces are the solid waste material of the human digestive system, which includes the bacteria. Appearance of fecal matter varies significantly in appearance. This difference is due to the state of the digestive system, the diet and the general health of the individual. Fecal matter is normally a solid waste with a mucus coating. Distal end might have pieces of the harder and less moist feces due to incomplete bowel movements and absorption of water. Human feces together with the human urine are collectively referred to as the human waste or the human excreta. Containing human feces and preventing the spreading of pathogens from the human feces via the fecal oral route are the main goals of sanitation. Sweat glands. In humans, eccrine glands secrete water-rich secretions which result in sweating. Sweating is a means of thermoregulation. An adult can secrete up to 2 to 4 liters of sweat per hour to 10 to 14 liters per day. Sweating is comparatively less in case of children before attaining puberty. Due to evaporative cooling, the evaporation of sweat causes a cooling effect. The two types of glands are the eccrine and the apocrine glands. The eccrine sweat glands. These glands are present since birth and are present all over the body, but its intensity is more in the plantar and the palmar surfaces. They produce water-based, clear and odorless secretions which results in cooling in humans. Salt is present in sweat, which is reabsorbed by the duct to lessen the loss of salt. In case of cystic fibrosis, this is the dysfunctional leading to production of salty sweat. The function of these glands is controlled by the autonomic nervous system that responds to thermal, emotional and medical stimuli. These cannot be consciously controlled by the individual. They are active in thermoregulation by cooling the body by sweat evaporation. After evaporation, the white sediment type substance is left due to the increase in the concentration of salts present in the sweat. The skin is one of the routes used by the body to eliminate the waste products. It is also used as a method of cooling the body through the evaporation of sweat. These two functions are combined as the sweat carries the waste materials to the surface through the eccrine glands, where they are washed away as we bath. Since there are limited amounts of waste materials to be discarded, the quantity exuded out at any time depends upon the opportunity the body has had to remove them. For instance, in cool weather, the body does not become overheated as frequently in warm weather, so there is no need to sweat much to provide cooling. Waste products are therefore not eliminated very frequently, but when sweating does eventually occur, the body uses the opportunity to excrete relatively large amounts of waste. Thus, the concentration in the secretion temporarily becomes very high. Conversely, in the summer, when the body needs to be cooled, sweating is frequent and often profuse, but there is still only the same amount of waste material to eliminate, because the opportunity to remove the material is almost constant. The concentration of waste materials at any particular moment is therefore much less. Composition of the eccrine sweat. Because many of the variables involved, the authorities vary slightly with respect to the exact proportions, but are generally agreed that the composition of the eccrine sweat is approximately water, that is 98.5% to 99.5%, solids present in 0.5% to 1.5%. These solids are approximately 1/3 to 1/2 of the inorganic salts and half to 2/3 of the various organic substances. Inorganic salts. The chloride, bromide, iodide and fluoride form the inorganic salts, sodium chloride being the most abundant. The concentration of the salts in the sweat is subjected to the factors that I am going to tell you about. First is the rate and the duration of the sweating. Next is the intermittent versus the continuous sweating. Third factor is the diet, and fourth is age and the sex. Organic substances. Of the many organic substances present in the eccrine secretions, those of significance in the identification work includes the amino acids and the fatty acids. Also present are the ammonia and urea. These materials are generally water soluble. The amino acids. Many amino acids have been successfully identified in normal eccrine secretions. Under conditions of normal daily activity, the concentration of the amino acid decreases with high rates of sweating in proportion with the other materials. One very big exception in that is the autonomic nervous system that causes the body to secrete very large amounts of amino acids when it is placed under stress. The autonomic system is the one that controls the heart rate and the other bodily functions over which we normally have no direct control. Fatty acids, these naturally occurring fats and oils are found within the group of substances known as lipids. The lipid group also contains such substances as waxes and squalene that are found in the sebum. It is important to understand that the oils and the fatty acids do not remain in a viscous state, as does motor oil, for example. The lipids are changed over a period of time through oxidation and hydrolysis. This is sometimes referred to as the drying out. It would be perhaps better to use the term cured just to differentiate the process from simple evaporation of substances such as water, urea and ammonia. Ammonia and urea are also present on the skin in quantities that vary with the rate of sweating. Apocrine sweat gland. These glands are present in the armpits, mammary, perianal and the genital areas in humans. Modified apocrine glands include the ciliary glands in the eyelids, the ceruminous glands which produce the earwax and the mammary glands which produce milk. In humans, they do not have any significance in thermoregulation. The apocrine glands are the sole effective sweat glands in hoofed animals like the horses, donkeys, etc. These are present since remain but inactive till the puberty. They are restricted to hairy areas of the body as they open and secrete into the hair canal. The presence of these glands in more of the Asians as compared to the Caucasians, women have more apocrine glands as compared to that of men. They have short ducts which lie close to the hair follicles. Non-primate mammals have apocrine glands in their larger parts of the body. Dogs and cats have acrine glands only in their foot pads and snout. Each hair follicle of these animals is covered with the apocrine sweat glands, an odorless, oily, opaque secretion that gains its characteristic odor upon the bacterial decomposition is produced similar to that of the humans. Presence of acrine glands on their paws increases the friction and prevents them from slipping. Composition of the apocrine sweat. These glands secrete an oily, odorless fluid containing lipids, proteins and steroids. These secretions are mixed with sebum as apocrine and the sebaceous gland which produces the sebum lie close to the hair follicle. In marsupials and hoofed animals, the acrine glands are the main thermoregulators which secrete water-based sweat. In most of the mammals, the apocrine glands secrete oily and smelly substances which act as pheromone, warning signal and the territorial marker. Since it is sensitive to adrenaline, it is involved in emotional sweating in humans. During the gestation period of the fetus, that is 5-month-old fetus, having the apocrine glands distributed all over the body, but it gets restricted to only few areas after few weeks. These glands secrete their secretion in the periodic spurts in contrast to the acrine glands. These glands were originally thought to use only the apocrine secretions, that is, the vesicles pinch off the secretory cells, then degrade in the secretory lumen and releasing their product. But recently, it has been shown that the eccrine secretion also takes place. A smooth muscle lining of the myoepithelial cell are formed around the secretory cells. After contraction of the muscles, the secretory ducts are squeezed and then accumulated fluid is pushed out into the hair follicle. Mixing of sweat and sebum takes place in the hair follicle, and then the mixed fluid arrives at the epidermal surface. The sweat produced by this gland is cloudy, viscous and have a pH of 6 to 7.5. Sodium chloride, protein, water and carbohydrate waste materials is present in it. After being degraded by bacteria, a specific odor of the sweat is produced and released. More the number of the bacteria, the stronger will be the odor. The odor becomes more pungent due to presence of hair as the secretions, keratin, debris and bacteria accumulate on the hairs. Various characteristics of the fecal matter. So under this we have first the color. Depending upon the diet and health of the individual, the appearance of the human fecal matter is different. First is brown color. It is a light to dark brown coloration due to the combination of the bile and the bilirubin which is derived from the dead red blood cells. It is covered with the mucus and is semi-solid. Yellow. Due to the parasitic infection like giardiasis, feces can be yellow in coloration. Giardiasis is caused by giardia, which can cause severe and communicable yellow diarrhea. Gilbert's syndrome can also be one of the reasons for the yellow coloration. If the food passes through the digestive tract very quickly, then it can lead to yellow stool. Yellow stool can also be found in people with GERD, that is gastroesophageal reflux disease, pale or grey. Due to insufficient bile output conditions such as the gallstones, cirrhosis, cholelithiasis, etc. can cause pale or grey stool. Liver produces bile salts which gives brownish color to a stool. Color of stool becomes light due to the decrease of the bile output, black or red. In case of the red blood cells have been in intestine for long enough to be broken down by the digestive enzymes, then feces can be of black color. This condition is known as melena. It can be due to bleeding in the upper digestive tract. Blood in stool can be due to other reasons also, such as the colon cancer and fissures and hemorrhoids, etc. The same color change can be observed after consuming foods that contain a substantial proportion of the animal blood, such as black pudding. The black feces can be because of some medications such as foods or dietary iron supplements also. Hemoptysia is similarly the passage of feces that are bright red due to the presence of the undigested blood either from the lower in the digestive tract or from a more active source in the upper digestive tract. The blue color. Prussian blue or blue, a coloring used in the treatment of the radiation, cesium and thallium poisoning, can turn the feces blue in color. In case of the food consumed contains blue dye, then it can also have the same effect. Violet or purple. Violet or purple feces is a symptom of porphyria. Green color. Large amount of the unprocessed bile in the digestive tract and strong smelling diarrhea can make feces green. Sometimes similar effect can also be seen due to the consumption of licorice candy made from anise oil and sugar rather than the licorice herb. Excessive sugar consumption and sensitive to anise oil can also cause green loose stools. Then silver. A tarnished silver or aluminium paint-like feces color characteristically results when biliary obstruction of any type, that is white stool, combines with the gastrointestinal bleeding from any source, that is black stool. The gastrointestinal bleeding and biliary obstruction can be due to the carcinoma of ampulla of water, which will in turn result in silver stool. The odor. Health and diet of a person can vary the physiological odor of feces. Hydrogen sulfide is the most common volatile sulfur compound in feces. The odor of feces may be increased in association with the various pathologies including the celiac disease, Crohn's disease, ulcerative colitis and malabsorption, etc. The odorant volatiles are responsible for odor of the human feces. The benzopyrrole volatiles such as the indole, acetone and hydrogen sulfide. Next is the methyl sulfides, wherein we have the methyl mercaptan or the methanethiol, that is MM, dimethyl sulfide, dimethyl trisulfide and dimethyl disulfide. The chemical composition. About 128 grams of the fresh feces is excreted by a person per day with a pH value of about 6.6. It contains 75% of water and remaining solid portion in about 84 to 93% organic solids. These organic solids contain 2 to 25% of the nitrogenous matter of protein, about 25 to 45% of the bacterial biomass, 25% carbohydrate or the undigested plant matter, and 2 to 15% undigested fat. Diet and body weight are some of the factors on which these proportions may differ. The calcium, intestinal secretions, little amount of dried epithelial cells, iron phosphates and mucus are the components of the remaining non-organic solids. Sweating is a process in which it produces fluids and these are secreted by the sweat glands in the skin of mammals. This process is also known as perspiration or diaphoresis. Fecal matter or the human feces are the solid waste material of the human digestive system, which includes the bacteria. Sweating is a means of thermoregulation. Eccrine glands are present since birth and are present all over the body, but its intensity is more in the plantar and the palmar surfaces. The eccrine glands produce water-based, clear and odorless secretions which result in the cooling in humans. The composition of the eccrine sweat is approximately water 98.5% to 99.5%, solids 0.5% to 1.5% and the solids approximately about 1/3 to half of the inorganic salts and half to 2/3 of the various organic substances. The apocrine glands are present in the armpits, mammary, the perianal areas and the genital areas of the humans. The apocrine glands secrete an oily, odorless fluid which contains lipids, proteins and steroids. These secretions are mixed with sebum as apocrine and the sebaceous gland that produces sebum lie close to the hair follicle. During the gestation period of fetus, that is 5 months old fetus, having the apocrine glands, they are distributed all over the body and it gets restricted only to few areas after few weeks. Depending upon the diet and health of the individual, the appearance of human fecal matter is different. Hydrogen sulfide is the most common volatile sulfur compound in feces. About 128 grams of fresh feces is excreted by a person per day with a pH value of about 6.6. Feces contain 75% of water and the remaining solid portion makes up to about 84 to 93% of the organic solids. These organic solids contain 2 to 25% of the nitrogenous matter or protein, about 25 to 45% of the bacterial biomass, 25% of carbohydrate or the undigested plant matter and 2 to 15% of the undigested fats.